This video will talk about the cell cycle, more specifically the eukaryotic cell cycle. The cell cycle consists of 4 phases: G1, S, G2, and the M phase. The first 3 phases, G1, S, and G2, are part of interphase, an in-between stage before and after cell division. The length of each phase varies among different types of cells, but interphase generally is the longest, because cells won't be dividing most of the time. A cell will spend about 90% of its time in interphase, which is about 23 hours compared to the 1 hour that takes for mitosis to occur. The movement of cells through the cell cycle is well regulated. There are checkpoints after each phase to make sure that the cell is growing properly and everything is going according to plan. In fact, you can think of them as stoplights telling the cell when to stop and to go. The G in G1 and G2 stands for gap, but these phases are actually a time of major cell growth. Most of the cell's growth happens during G1, where cells get larger and synthesize new proteins and organelles. Keep in mind that cells can also leave the cycle and quit dividing. This can be temporary or permanent, and this phase is called gap 0. Otherwise, the cell will continue on into the S phase, or the synthesis phase. Chromosomes are replicated and new DNA is synthesized, doubling the amount of DNA in the cell. When DNA replication is finished, the cell will prepare for division, or the M phase, in the G2 phase. The G2 phase is where organelles required for cell division are produced. The M phase is where cell division occurs, and it takes place much faster than interphase. The M phase consists of 2 stages, and the first stage, mitosis, is the division of the cell nucleus. Mitosis is split into 4 phases: prophase, metaphase, anaphase, and telophase. The second part of the M phase is cytokinesis, and it is the division of the cytoplasm. Now, the first phase of mitosis, prophase, is where the chromatin condenses into visible chromosomes in the nucleus. This happens because the centromere links 2 chromatids together to form a chromosome. Outside the nucleus, spindle starts to form from the area where centrioles are located. The nucleolus fades, and the nuclear envelope breaks down. Metaphase is the second phase of mitosis, and it is the shortest. During this phase, the chromosomes align in the center of the cell, and spindle fibers connect with the centromeres of the duplicated chromosomes. The third phase, anaphase, is where sister chromatids separate and begin to move along the spindle fibers to opposite ends of the cell. The final step of mitosis is telophase, where the chromosomes arrive at the opposite poles and spread out into a tangle of chromatin. The nuclear envelope forms again around the 2 tangles of chromatin. After mitosis is completed, cytokinesis basically finishes the job, splitting one cell into 2 and dividing the cytoplasm. The result is 2 daughter cells that each will undergo interphase, both continuing the cell cycle. The cell cycle isn't always successful, and it is because of 2 types of genes that play a major role in regulating the cell cycle. Proto-oncogenes encourage cell division, while tumor suppressor genes do the opposite. The success rate of the cell cycle depends on the balance between these 2 genes. If mutations form in these genes, tumor suppressor genes become inactive, while proto-oncogenes become oncogenes, causing cells to divide and grow uncontrollably, which can lead to cancer. These mutations can happen anywhere in the cell cycle. Other things like smoking, drinking alcohol, family history, and sun exposure can all increase your chances of getting cancer.